Exercise doesn't just burn calories, it sends messages, literally. When you exercise, your muscles release billions of tiny packages called extracellular vesicles, or ECVs. These are nano-sized bubbles filled with proteins, microRNAs, and signaling molecules. Think of them like biological text messages that your muscles send to your liver, your fat, your brain, and even your immune system. And the message is powerful. Exercise-induced ECVs improve insulin sensitivity, reduce inflammation, boost mitochondrial function, and even help turn white fat into calorie-burning brown fat. Studies show these vesicles actually deliver instructions that reprogram how other tissues behave. That's why exercise helps your whole body, not just the muscles you're using. So when you work out, you're not just moving. You're changing the molecular conversation inside your body. That's exercise as medicine. This is lecture 136 of the Metabolic Classroom. Welcome back to the Metabolic Classroom. I'm Ben Bikman, metabolic scientist and professor of cell biology. Today's mini lecture aims to teach you about one of the most exciting frontiers in exercise physiology, namely extracellular vesicles, or what I will just abbreviate as ECVs. Sometimes it's abbreviated as EVs, and I just want to make sure that everyone's clear that I'm not talking about vehicles, so I'll say ECVs. If you've never heard of extracellular vesicles, you're not alone. This is a relatively new area of research that's really revolutionizing how we understand cellular communication, and it has profound implications for why exercise is so beneficial for virtually every system in your body. Think of ECVs as tiny biological packages that your cells send to each other. They're nano-sized bubbles, if you will. They're far, far smaller than the cells themselves, and they're carrying molecular cargo. So that means that these little packages are carrying proteins and lipids and even RNA and DNA. When you exercise, your muscles, fat tissue, liver, and other organs ramp up production of these vesicles and release them into circulation, delivering instructions that can change how distant tissues will function. This is a fundamentally different way of thinking about how exercise works. We're not just talking about burning calories or building muscle. We're talking about your working muscles literally sending molecular instructions to your brain, your liver, or your pancreas, or even your immune system. But before we understand how exercise affects ECVs, I really want to make sure you understand what they are. Extracellular vesicles are membrane-bound particles, and the cells release them into the extracellular space. That's just the fluid, the environment right outside of the cells. There are several types of ECVs classified mainly by their size and how they're formed. The main categories include exosomes, which are typically in the order of 30 to 150 nanometers in diameter. And then there's also microvesicles, which are actually larger at around in the range of several hundred up to 1,000 nanometers. There are also what's called apoptotic bodies, which are released when cells die. But for today's discussion, we're focusing primarily on exosomes and microvesicles, which are the most studied in the context of exercise. Here's what makes ECVs so special. They're not just random debris that cells are shedding. They're carefully constructed packages with specific cargo. The parent cell loads these vesicles with particular proteins and lipids and, as I noted a moment ago, nucleic acids, especially microRNAs, which are small RNA molecules that can regulate gene expression in the cells that are receiving the message or receiving the package. The vesicle membrane itself is also critical. It's studded with proteins that act like address labels. That helps the ECV find its target tissue. This targeting specificity is remarkable. A vesicle released from your skeletal muscle during exercise might have molecular markers that direct it specifically to your liver cells or your adipocytes or even your brain. Once an ECV reaches its target, it can deliver its cargo in several distinct ways. Sometimes it fuses directly with the target cell's membrane, releasing its contents inside. Other times, the target cell engulfs the entire vesicle through endocytosis. Either way, the molecular messages get delivered, and the recipient cell's behavior can change dramatically and sometimes immediately. This is why ECVs are sometimes called the body's molecular mail service. But unlike email, which carries just information, these vesicles can carry functional molecules that immediately start working in the recipient's cell. So if it is a mail service, it's delivering packages, something far more substantial than, say, just a small letter or let alone an email. Now, here's what makes this relevant to metabolism and health. Virtually every cell type in your body produces ECVs, and every cell type can receive them. Your adipocytes release ECVs. Your hepatocytes release ECVs. Your myocytes release them, and even your neurons. It's a constant conversation happening throughout your body, and exercise dramatically changes both the volume and the content of that conversation. So what happens to extracellular vesicles when you exercise? The short answer, a lot. In 2018, a great study, in fact, the study was published in Cell Metabolism, and it changed how we think about exercise. These researchers had healthy individuals perform 1 hour of cycling exercise and then used advanced proteomic techniques to analyze what was happening in their blood. They found that exercise increased the circulation of over 300 proteins, with notable enrichment of proteins that compose exosomes and small vesicles. Using imaging experiments, they showed that these exercise-induced ECVs actually homed in on the liver, and then they were able to transfer their cargo, these proteins, to the liver cells. This was a paradigm shift. It wasn't just that exercise releases signaling molecules. Exercise triggers a massive release of membrane-bound packages that can deliver entire molecular programs to these distant organs. But it's not even just about the quantity. The quality of the cargo was also quite remarkable with regards to these vesicles. These exercise-induced ECVs carry a distinct molecular signature compared to ECVs that are produced during rest. They are enriched with specific microRNAs that promote many beneficial metabolic changes and anti-inflammatory proteins and even growth factors. Specific types of exercise produce different ECV profiles. For example, aerobic exercise tends to generate ECVs that are particularly rich in microRNAs associated with angiogenesis, that's the formation of new blood vessels, as well as metabolic regulation. Whereas resistance exercise produces ECVs with higher concentrations of myokines, which are muscle-derived signaling molecules or hormones, as well as proteins involved in muscle growth and repair. But also the intensity matters. Research suggests that high-intensity interval training produces a more robust ECV response compared to moderate continuous exercise, at least in terms of the acute increase in ECV numbers, as well as the concentration of beneficial cargo. One of the most fascinating aspects is that exercise-induced ECVs appear to carry the memory of the exercise stimulus to other tissues. For example, ECVs released from contracting muscle can carry metabolic signals to fat tissue, essentially instructing your adipocytes to increase their metabolic activity and become more insulin sensitive and increase greater degrees of lipolysis or fat breakdown. Recent research has also revealed something remarkable about mitochondrial function. A paper published just last year in 2025 showed that when muscle cells are electrically stimulated to contract, so a laboratory method of stimulating exercise, the ECVs they release can actually enhance mitochondrial biogenesis in recipient cells. These ECVs increased oxygen consumption rates and ATP production in muscle cells that received them. The effect was dose-dependent and was mediated by proteins on the ECV membrane. Essentially, active muscle cells can share their metabolic fitness with other cells through these ECVs. Another compelling finding involves the brain. While we've long known that exercise benefits cognitive function and mood, we're now learning that ECVs play a role. A 2024 study in older adults with prediabetes found that just 2 weeks of exercise training altered insulin signaling proteins in neuronal extracellular vesicles. So these are ECVs that originate from brain cells. The changes in these neuronal ECVs correlated with improvements in peripheral insulin sensitivity, suggesting a bidirectional communication between the brain and metabolic tissues via ECVs. Now, let's talk about how the ECV story or lesson connects to metabolic health, which of course is really the core focus of this podcast. One of the most significant roles of exercise-induced ECVs is in improving insulin sensitivity, I would submit. A 2022 study in adults with obesity found that a single bout of aerobic exercise decreased both fasting and insulin-stimulated extracellular vesicles. More importantly, the degree to which exercise reduced certain platelet-derived ECVs correlated directly with improvements in metabolic and insulin sensitivity, In other words, the people whose ECV profiles changed the most with exercise also showed the greatest improvements in how well their bodies responded to insulin. The mechanism appears to involve several pathways. Research using advanced microRNA profiling has identified specific microRNAs in exercise-induced ECVs that are associated with insulin sensitivity and adiposity. For instance, studies have found that MIR, or microRNA 6523P, and several other microRNAs in plasma ECVs change with exercise and correlate with insulin sensitivity as measured by the gold standard hyperinsulinemic euglycemic clamp. These microRNAs can influence insulin receptor expression and enhance insulin signaling pathways in target cells. ECVs also play a fascinating role in the browning of white adipose tissue. Brown adipose tissue, or brown fat, is very metabolically active. It burns calories to produce heat through a process called thermogenesis. Most of our fat on our bodies is white adipose tissue, which primarily stores energy. But it turns out that white fat can be converted to a more brown-like metabolically active state, a process called browning. And exercise-induced ECVs are key mediators of this process. Research has shown that long-term exercise leads to changes in EV-associated microRNAs that promote fat burning and fat browning. Specifically, exercise reduces levels of a microRNA called 191A5P in circulating ECVs. When this microRNA is suppressed, it allows increased expression of a gene called PRDM16, which is a master regulator of the browning process in fat cells. This then, in turn, leads to increased expression of thermogenic proteins like uncoupling protein 1, UCP1, which enhances heat production, and of course the net effect of all of this would be reducing obesity. The uncoupling proteins is a topic I've discussed abundantly in the past, so if you need to review, please do so by looking through the previous Metabolic Classroom episodes. There's also compelling evidence that the myokine irisin, which is released from exercising muscle, can be packaged into extracellular vesicles. A 2024 study found that during sustained exercise, irisin accumulates in circulating ECVs, and these vesicles bind irisin molecules within them, and they in turn can promote white fat browning by activating AMPK signaling and enhancing adipose energy metabolism. In fact, a rodent study found that when as they tracked this, the animals lost significantly more weight, and indeed they found much greater degrees of thermogenesis, or uncoupling or fat burning in the fat cells. From an inflammation standpoint, exercised ECVs are generally anti-inflammatory, which might seem paradoxical because exercise itself is an acute inflammatory stimulus. The resolution of that paradox, I think, is a matter of timing and context. While exercise causes acute inflammation in the working muscles, the EVs released during and after exercise carry anti-inflammatory signals that help resolve inflammation systemically. These ECVs contain microRNAs that suppress pro-inflammatory cytokine production and proteins that promote what's called M2 macrophage polarization. That's the anti-inflammatory aspect of macrophages, when the macrophages are focused more on tissue repair rather than promoting systemic inflammation. This is one mechanism, I think, that can explain why regular exercise can reduce systemic inflammation, which, of course, we know is a major driver of insulin resistance and the metabolic consequences that follow. Now, it's important to understand that not all ECVs are beneficial. In metabolic disease states, the ECV landscape changes dramatically and often for the worse. In obesity, for example, adipose tissue releases ECVs with an altered cargo profile. These ECVs are enriched with pro-inflammatory molecules, microRNAs that promote insulin resistance, and lipids that can induce cellular stress. Essentially, dysfunctional fat tissue is sending harmful messages throughout the body via ECVs. One particularly striking study showed that when ECVs from adipose tissue that were isolated from individuals with obesity, and then it's a preclinical study. So they took the ECVs from adipose tissue of obese mice and injected them into lean mice. The lean mice developed glucose intolerance and insulin resistance. Conversely, when they took ECVs from lean adipose tissue and gave them to the obese mice, it restored glucose tolerance and insulin sensitivity. And they were able to identify a very specific microRNA, and they found that it was overexpressed in obese fat tissue ECVs, and it then in turn was responsible for impairing the insulin action and all the other consequences that I noted. This simply demonstrates something quite remarkable. ECVs aren't just correlated with metabolic disease, they can actually transfer metabolic dysfunction from one individual to another, at least in the animal model. Another way of looking at this could be that it's capable of passing the metabolic dysfunction from one tissue to another, in a bit of a bizarre twist of how exercise is able to pass the benefit from one tissue or one cell to another. In type 2 diabetes, circulating ECVs show distinct abnormalities, so the problems can stack up. Research has found that people with type 2 diabetes have different ECV microRNA profiles compared to those with normal glucose tolerance. For example, a specific exosomal microRNA is highly abundant in individuals with type 2 diabetes and can modulate insulin-stimulated glucose metabolism through effects on the AKT signaling pathway in skeletal muscles. That AKT pathway is just one of the critical downstream effects of insulin. So I say AKT signaling, I could have just said something downstream along the insulin pathway. The liver, of course, is so important to everything involved in metabolism, and it's no exception here when it comes to ECVs. In non-alcoholic fatty liver disease, liver cell-derived ECVs carry messages that promote further fat accumulation, fibrosis, and inflammation. These ECVs can affect distant organs, contributing to systemic metabolic consequences. But here's where the exercise story becomes, I think, even more compelling. Regular physical activity can normalize the ECV profile even in instances of metabolic disease. Studies in people with obesity and dysglycemia, or just altered glucose levels, show that both acute exercise and exercise training over time shift their circulating ECVs toward a healthier composition, more anti-inflammatory signals, more insulin sensitizing microRNAs, and beneficial changes in ECV protein cargo. This represents a fundamental shift in how we think about exercise as medicine. We're not just talking about weight loss or improved cardiovascular fitness. We're talking about changing the molecular conversation that's happening between your organs and the cells of those organs at that very level, literally the reprogramming the messages that are coming from one cell to another. I think there are some practical implications of this. And of course, that's something I always want to try to highlight when I can to help the science come to life, if you will, a practical application. All right, so what are those? First, I think these ideas, this information reinforces that exercise, in my view, is not optional for metabolic health. Exercise is essential. The benefits of exercise really extend far beyond the muscle that you're actually working. Every time you are contracting and relaxing those muscles, you are triggering a systemic communication network through these ECVs, the result of which will be a benefit to your entire body, sending the signal to your liver and your fat, your immune system, your pancreas, and even your brain. Second, by way of implications, I think this information suggests that consistency matters tremendously. The ECV response to exercise is relatively short-lived. Studies show that ECV microRNA profiles return toward baseline within just hours after acute exercise. This means that to maintain the beneficial effects of the exercise-induced ECVs, you need to exercise regularly. The molecular conversation will die out unless you keep it alive. The good news is that you don't need to exercise for hours to get a meaningful ECV response. Even a relatively brief bout of exercise can trigger significant changes. Of course, high-intensity training appears particularly effective at generating robust ECV responses. And much to everyone's delight, I think, both aerobic and resistance exercise can produce beneficial ECVs. But there are some important differences when you look at the cargo profiles. This supports the recommendation to include, if you can, both types of exercise in your routine, because after all, you're going to be generating different types of molecular messages that complement each other. For people with metabolic dysfunction, things like obesity or insulin resistance, type 2 diabetes, or fatty liver disease, I think the ECV story provides both an explanation and, I would hope, some extra motivation. Your dysfunctional tissues are currently sending harmful messages throughout your body via these ECVs. These ECVs are, in a real sense, spreading the metabolic disease from tissue to tissue. Exercise is one of the most powerful tools you have to reverse that conversation, shifting toward beneficial health-promoting signals that can restore the metabolic function systemically and counter that signal that's coming from the sick cells. It's also worth noting that while I've focused on exercise, diet also can influence ECV landscape as well, though this is less well studied. We know that metabolic state affects ECV composition, and since diet is a primary determinant of the metabolic state, it stands to reason that what you eat is going to influence the molecular message that your cells send to each other. This is going to be an area of future research that frankly excites me a lot, and I expect a lot of fascinating effects and findings coming out in the near future. And the future is bright because when it comes to ECVs, the field is still relatively young. And of course, even younger and more less well known when it comes to understanding the role of exercise. But I think there are some exciting developments. Researchers are exploring whether ECVs could serve as biomarkers for metabolic health and exercise. By analyzing someone's circulating ECVs, we might be able to assess their metabolic state and get an idea of where they're at currently and what their risk for disease might be. This could allow for much more personalized exercise prescriptions. There's also interest in therapeutic applications, of course. Could we harvest ECVs from exercised individuals and use them as a treatment for metabolic disease? Animal studies have shown that transferring exercise-induced ECVs to sedentary animals can confer some of the metabolic benefits. And of course, we're a long way from clinical applications in humans, but even still, the concept that you could have some kind of exercise in a bottle or an injection when it comes to ECVs, it is being actively investigated. Some researchers are even exploring whether we could engineer cells to produce ECVs with specific therapeutic cargo, thereby creating designer vesicles that mimic or enhance the beneficial effects of exercise and even specific exercises. That could potentially help people who are unable to exercise due to perhaps disability or even illnesses. Another frontier is understanding some of the individual variation that comes along with this. Why do some people show robust ECV response to exercise while others show very modest responses? Could things like genetic factors or the training status or age and sex and metabolic health or other variables explain these differences? And can we use someone's baseline ECV profile to personalize their exercise recommendations? All of these questions and many more are actively being investigated. In my mind, as we wrap up, the lessons of extracellular vesicles and exercise reveals just how sophisticated the body really is. Exercise is a complex communication event where working muscles and active organs are metabolically engaged and in sending and preparing and packaging and sending these signals throughout the body. And in so doing, they're collectively coordinating many beneficial adaptations in tissues that might be far removed from the muscle that's actually doing the work in the moment. But your muscles are talking. They are sending these messages to other tissues and cell types throughout your body, and doing so with these remarkable nano-sized packages that are called extracellular vesicles. This is, I think, one of the most elegant examples of metabolism in action, of mediating and sending messages from working cells to cells that you wouldn't have imagined would be benefiting. I hope this is giving you proper and sufficient motivation, and I hope it's something you're going to remember next time you consider exercising. Keep in mind that you are doing more than just moving your body. You are triggering the release of billions of tiny packages that are going to travel through your bloodstream, find the specific target tissue, and deliver instructions to overall just promote significantly greater metabolic health. Thanks for listening to this mini lecture in the Metabolic Classroom. Remember, more knowledge, better health.